Hi, my name is Hawley Helmbrecht. I'm a Ph.D. student at the University of Washington in the Department of Chemical Engineering, and I have the great pleasure of doing my research in the Disease-Directed Engineering Lab of Elizabeth Nance. I'm also very excited to be giving this talk at the AiChE annual meeting in the Applications of Data Science to High Throughput Experimentation section on a project I've been working on called FFiber, or a framework for fluorescent neuroimaging-based experimental routines. And since I am giving a talk in the Data Science to High Throughput Experimentation section, I thought that I would start this talk in reference of an experiment we recently published in Bioengineering and Translational Medicine. So if you couldn't tell from the title of neuroimaging, in the Nance lab, we study the brain. And when we choose to start an experiment in the brain, we typically choose a disease model we're interested in studying, a cell or a few cells that we're interested in learning more insight about, and an animal model in which to do it. And I said that I was going to give this talk in reference to an experiment we recently published in Bioengineering and Translational Medicine. So I'm going to give a brief description of the methodology. So for us, we chose a rat model in order to do this study of oxygen glucose deprivation in ex vivo slices. And so what that means for ex vivo slice models is that instead of taking the entire brain as one model, we take the brain and we slice it on a tissue chopper, and each individual slice is a different treatment group. And so those slices can fall into three groups. They can be the non-treated control, the injured group, which undergoes half an hour of oxygen glucose deprivation, or injury plus treatment with azithromycin. And what is also important to know about our lab is that we obtain almost all of our data through fluorescent imaging. Additionally, for this paper with these three treatment groups trying to understand oxygen glucose deprivation in ex vivo slices, it took our lab 9 independent researchers that train or work with 17 undergrads, 7 high school students, and have trained, worked with, or used the materials or equipment of 9 collaborators across 3 institutions and 5 individual departments. Another really important thing to know about the nature of our research is that the Nance lab completes our research on the brain in 4 main areas. 1, we develop the slice culture models such as the ex vivo oxygen glucose development model described before. 2, we characterize brain injury, which is where most of my work falls. 3, we engineer nanoparticles and nanotherapeutics so that we can treat brain injury that we've characterized. And 4, we measure transport in live tissues so that we can get an understanding of how nanotherapeutics and particles are interacting with the greater brain microenvironment. And since we obtain most of our data through fluorescent imaging, it's important to know that our image procurement leads to large amounts of data. When you also include things like the treatment groups we have, the sex differences and age differences in animals, as well as regional differences within the brains themselves, and the fact that all of these need to be imaged, we get easily greater than a terabyte of data per experiment. And my own personal work that studies shape features of cells, some example shape features can give us over 50 features per cell segmented. And then we have to break down what do these example shape features such as area and perimeter and circularity mean in greater respect of the brain microenvironment. So to effectively perform experiments, we needed a framework to optimize our current methodologies while increasing our analysis capabilities. And that's why I created FFiber, or Frameworks for Fluorescent Neuroimage-Based Experimental Routines, which is a 7-step process that allows us to perform these experimental procedures in a high throughput manner. And these 7 steps go all the way from developing a data awareness to then training the domain scientists on the data science pipelines we've just created. So to start, I want to delve a little bit deeper into what we mean by developing a data awareness. Well, for us, this has been a 4-step process. Number 1, we rethink what data means. We sit down as a group or as subgroups or with our collaborators, and we really outline what data is most important to us. Is it only our results, or is it also the people that get those results? Is it also all of the chemicals that are used? Is it also all of these details about the animals and their behavior? Once we rethink what data means to us, we perform an assessment of the relevant metadata. That way we understand all the factors that we need to keep track of in order to really have an efficient experimental program. Thirdly, we identify appropriate data storage locations for both temporary and terminal data storage. That way we know exactly where our data is going, what file types it's going to be in, and where it's going to end up. And finally, we make connections between data relationships. This is very important, especially in our ex vivo slice culture models, where one slice can go to an specific experiment and another slice can go to a different experiment, that we understand how all of these things relate and correlate. After we've developed a data awareness, we design a data management plan. But an effective data management plan is a holistic process to be consistently updated and specifically implemented. So what I mean by that is that our data management plans aren't created on an experiment by experiment basis, but actually on a quarterly basis. So what we do quarterly is data management workshops and worksheets so that all of our researchers, from our graduate students to our high school students, have worked on a data management plan. And then we actually create these preliminary data management plans for all techniques and methodologies that are commonly used throughout the lab. And we follow up with regular data storage assessments, so we're understanding of how much data we're actually generating, where it is, and what forms and file types it is included in. Here's an example of a preliminary data management plan from our research technician, Kate Hildahl. And this was one she created before we ever completed the oxygen glucose deprivation experiment. But because this plan had already been used, we can specifically implement the more detailed version of this plan straight into the experiment so that we already had a data management plan going in. After we develop a data management plan, we determine an optimal experimental pipeline. Optimal experimental pipelines for neuroimaging work with large amounts of image processing necessitate both domain and data science expertise. What we mean by this is that it's important that we have our experimentalist expertise to understand how the pictures are taken and how that actually relates to the biological data that they have themselves procured. But it's also important that our data scientists bring their expertise to the table so that we know which features are actually feasible to add on and what analysis techniques and what things we want to analyze which are actually possible. On top of that, for our experimentalists, we address their labor hour bottlenecks, we identify their output data types and file structures, and we identify personnel responsibilities for an effective experimental pipeline. And for our data scientists, we also address labor hour bottlenecks, we perform software reviews, and we identify necessary programs, packages, or scripts that we may have to build out or develop ourselves in order to create our analysis. After we have an optimal experimental pipeline, we build out the supporting data science infrastructure. Within the Nance lab, we've applied this cell morphology process enough times that we have a standard pipeline. And so all we have to do is adjust for our specific experiment and allow for our work to go through this pipeline, through our thresholding techniques, our shape analysis, and then finally to data visualization of our results. So once we build out the data science infrastructure, we have our experimentalists actually perform the imaging. What's amazing about FFiber is that our experimentalists already know who's doing what, where it's going, how much data it's going to be, and our data scientists already know how many images are being procured and in what file types, so we can go straight into getting results and then producing interpretable visualizations. So at this point, I'd like to give a more specific example of how we've applied FFiber in two areas. FFiber is experimentally agnostic, so it can be done both initially at the beginning of an experiment and retroactively. So I'm going to show you an example of how we used FFiber initially at the beginning of the oxygen glucose deprivation model experiment that I just explained. So for the oxygen glucose deprivation model, what I had for our pipeline were images of these three treatment groups: the non-treated control, that injured oxygen glucose deprived group, and then the treatment group. And the hypothesis that I was looking to answer is that oxygen glucose deprivation induces detectable phenotypic changes in microglia that provide insight into the greater brain microenvironment. So I took those example shape features that I talked about earlier, ran that on all of our cells, which is thousands of cells, and then we found something significant in circularity, which was exciting. We found that there's a significant difference between our non-treated control and our injured group, that the injured group shows more circularity in the cells that is then reverted back to be more similar to the non-treated group with the azithromycin treatment after injury. This is exciting because it correlates with phenotypic changes in microglia that say with injury and with death, microglia change to a more amoeboid and circular state. Alongside doing our circular analysis, we perform a vampire analysis, which is visually aided morphophenotyping image recognition from the Dennis Wart's lab at Johns Hopkins. What we do there is we take our segmented cells and basically take coordinate points on the outside of these cells, split them into training and testing groups, and and have our training groups build a model of a dendrogram of representative phenotypes, and then apply that to our testing group so that we can see how frequent those phenotypes are in each of our treatment groups of interest. When we did this, we found an interesting difference that there is a difference in representative phenotypes between our non-treated control and our OGD group, which is then changed and reverted slightly with treatment. Looking at this in another way, in terms of microglial heterogeneity, we actually can find that there is less heterogeneity in the injured group. That being said, all of their representative morphological phenotype frequencies are more similar, and so there's higher heterogeneity in which shapes are existing in that system, and that is also reversed with treatment. So this was an example of how we can apply fiber initially at the beginning of an experiment, but we've also applied it retroactively in an E. coli-derived lipopolysaccharide inflammation model, where we were looking at how inflammation induces regional differences in microglial morphological heterogeneity. And by regional differences, we mean that we took the shape mode frequencies and applied the vampire process to 4 different regions of the hippocampus of Sprague Dawley rats. Those regions are the CA1, the CA2, the CA3, and the DG. And from there, we found that there was actually quite a lot of difference just between the DG and the other 3 regions within the hippocampus. This was interesting, and we applied it into a grant. We've also done the same application in age differences and found significant differences there as well. So all of this was made possible by fiber, but after we did the 6-step process into the oxygen glucose deprivation model, we realized we needed one more step, which is textile or domain science education, which are tutorials for experimentalist interactive learning. That way data scientists can pass on their knowledge of the pipelines they've created so domain scientists can run them themselves. We've actually already created 6 modules that we've run by 6 high school students, 4 undergrads, and 5 graduate students that basically pair learning of data science, such as a brief history of coding, the concept of what a data scientist is, and version control with specific image processing techniques that we're using in the Nance lab. That has allowed us to actually train our domain scientists in data science with minimal downtime. And then finally, fiber is a robust and iterable framework for designing and carrying out neuroimaging experiments. So every time we perform an experiment, we go back in and make fiber better. So after these experiments, we improved our first 3 steps. We realized that we also wanted to create databases of relevant image data so that we could increase the possibilities for more retroactive experiments. We're increasing our organization and documentation. That way we have a more efficient transition between experiments, and we're developing automation for labor hour intensive processes. Every time we iterate this process, we're able to break down some of those labor hour bottlenecks to free more time for our researchers. Finally, we assess data storage locations. Through our recent processes, we've been able to decrease most of our image storage costs to $0. Additionally, we recognize new packages that we'd like to build, and we started building a package called If Thresholds that allows us for better thresholding of cells in tissue along with quantitative scoring methods. On our 4th step, building out data science infrastructure, and our 5th step, performing primary and supplemental imaging, we have developed new packages and features that add on to our comprehensive cell morphology pipeline based off things our experimentalists have pointed out to be important features we're interested in studying in cells. And from producing interpretable visualizations of results, we have allowed us to increase our database of images and improve our interpretability and biological insight with greater connection between our other experimental procedures. But the future experimental applications of fiber are bright. We can apply it to alternative imaging types such as non-fluorescent immunohistochemistry. We can apply it on an array of neurological models, hopefully traumatic brain injury in the future and other inflammation models. And it doesn't need to be only applied on microglia. It can work on neurons, astrocytes, and oligodendrocytes as well. And we have future data science features we'd like to build out as well, such as the creation of metadatabases for easier data access and improved data connections, budget versus storage matrix. We're working with the computer science engineering program at UW for economic cost optimization and effective data access, and better documentation for automatic electronic lab notebooks and generalized processing pipelines. So in conclusion, fiber allows us to process data more efficiently while supporting greater insight into the brain microenvironment. I'd like to thank my lab, our funding, and my amazing PI, Elizabeth Nance. Thank you so much for allowing me to give this talk today.